Next question, we have molecule. What is this? This is quite common. This is double helix. Very obviously, this is our DNA. So please write the full name. Full name. Deoxyribonucleic acid. Deoxyribonucleic acid. And what does this belong to? What's the name of the family? What is the name of this whole compound? This compound is called nucleic acid. Okay, the family of it is called nucleic acid. Who's the element? CH or NP. CH or NP. Please don't write me short form. This is not chemistry. This is not chemistry. Your teacher cannot read. I mean, cannot, but they don't want to read CH or NP. They want to read the actual name. So please give the full name: carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay, please give the full name. Okay. Cool. Next one. The basic unit, no hard. Remember, this is what you also learn in your class easily. One circle, one pentagon, one square. I mean rectangle. And do remember that you must label your diagram, even if it doesn't say label. Any diagram must be labeled. And when you label it, please don't write inside the circle. Don't write inside the thing. You should draw a circle. You should draw an arrow out. So this is phosphate group. This is pentose sugar. This is nitrogenous base. Again, feel free to pause if you need to. Feel free to pause if you need to. Okay, so this is your nucleotide. This is, remember the name? This is your nucleotide. Now, with this nucleotide, you will now start to become a DNA. How can you form a DNA? Then we can see the next page. You can see it will become two chain. So let's complete the drawing. Remember how to draw the drawing? It must be flipped. Remember? It must be flipped. So remember your draw line. You can try to just draw the whole your whole paper. Just turn it around and draw like this. Turn the paper around and draw. And you see, try to match the shape. Try to match the shape. So like this, match it. Okay, try to match the shape. So make it a bit clear lah. Make it a bit clear. Okay. In this case, they only draw two. Can also. You see, they only draw two circles here. They also draw two circles. Can. So I just draw like one circle, another one like this. Again, I'm now drawing it the palette. I am now drawing it the palette. So my iPad is not the palette. So you should also do the same. You should also do the same. So you draw it the other way around. Okay. Again, try to fit the shape. Try to fit the shape. You see, this is basically the same as this. So later you will see, when you draw this one, it should also be like this, long a bit. Try to fit the shape. Okay. Try to fit the shape. Okay. And lastly, don't forget there should be a hydrogen bond. There should be a hydrogen bond. So I want you to see, I want to see you drawing the dot here. Okay, there must be a dot here. Usually you put two of them. If you want to make it very precise, actually it should be 2 and 3. So let's say this pair have 2 bonds, then the next pair should have 3 bonds. Again, this is very particular. I don't think they care about this, but if you really can do that, you can help me to do this. Basically, what I mean is that you can try to draw the number of bonds according to the pair. Just remember that one pair has 2 bonds, one pair has 3 bonds. But again, it's okay if you can't. It's okay if you can't. Just draw 2 and 3, whatever. Just draw 3 and 3. Okay, that should be enough. Okay, so that is it for this part. Okay, this is our DNA. You see, 1 line, 2 line together become DNA. Okay, next one. What is this big thing? This is chromosome. This big thing is called chromosome. And what do you do? What does it do in relation? You haven't learned about this. But you see, don't care whatever is this. Again, you have learned before. You learned already in chapter 4. You learned it already that this is a protein. Remember, we did mention before. Protein has a lot of example. Antibody, enzyme, hormone. There's a lot of example in which enzyme is one of them. Enzyme is also protein. Enzyme is also protein. So this is basically asking you how to produce a protein. So remember, what should you do? X will have what? X will carry DNA. I mean, in this case, actually, molecule W is DNA. Sure, let's use molecule W. This is DNA, okay? So molecule W is DNA. So X carry molecule W. And W is a DNA. That's a main character. That's a main character. What does DNA do? DNA will carry the genetic information. Genetic information for the enzyme synthesis. For the enzyme. Okay, remember your question was asking for enzyme. So yeah, if you want to know how can I produce this enzyme, you should ask DNA because DNA is the one that control all the information. So you're going to ask him how to do the DNA, how to do the enzyme, and then what happened? You will need to do two process. I mean, technically three, two. Okay, so yeah, first of all, you need to copy this out to your ribosome. Remember, this is called transcription. So the information here is transcribed. Okay, you copy them out. You copy the manuscript out to your Sejarah textbook. For example, you copy them out. This is called transcribe. It will be transcribed into mRNA. Remember this term? Our power trader. This this GrabFood driver, they will transcribe into this delivery man, and then this delivery man will then translate into amino acid. Okay, sequence of amino acid. Okay, it will then translate into the sequence of amino acid. And finally, there can then be condensation. Amino acid can bond together. They use a very weird word, but you can also say combine, condense, whatever, like bond together also can. They bond together to form the what? Okay. Remember the question? The question was asking for this. So yes, please go back to this to form the extracellular enzyme. Okay, to form the extracellular enzyme. Okay, that's it. Yep, and let's move on to next question. Okay, I hope this is still okay. But you see, basically just go through the whole procedure. Just go through the whole process. This is basically protein synthesis. Okay, this is basically protein synthesis. So remember, I do mention already before, you need to be able to recognize a few different examples of protein. If I tell you enzyme, you need to remember it's a protein. I tell you hormone, you need to remember it's a protein because we have a lot of things to do with protein. So do remember, there's few example of protein is inside nota. Okay, okay, that's it. Let's see question C1. Why and another type of molecule is another type of compound. So I mean this is actually RNA lah. Okay, so you have DNA, you have RNA, right? Okay, so compare and contrast DNA and RNA. This is not hard. This is not hard. So let's start from compare similarities. Okay, since you know lah, they are both under this chapter of DNA, of nucleic acid. So yeah, they are both nucleic acid. And that means they will have all the characteristics of nucleic acid. What do you know? For example, you know that nucleic acid is made up of the monomer, nucleotide. So yeah, not wrong. Both of them should have nucleotide because they are all nucleic acid. Okay, if both of you are student, you must have the similarity as a student. If both of them are DNA and RNA are nucleic acid, same thing, no? They must then have the characteristic of DNA. So you can branch it from there. Okay, what else? CHOMP. You should have CS or MP. I don't write the full name, you can write the full name yourself. CHOMP. Okay, yeah. And in fact, one more thing, this is a bit specific, but if you remember, you have three bases that is the same. You have three bases that is the same. These bases are A, C, G. These bases are A, C, G. So why not T? Why not T? Because later you will remember, because T is changed to U. If you remember that, in DNA we have T, in RNA we have U. That's why you have a bit of difference over here. But on top of that, I think it's all the same. Okay, yeah, that's why. That's why you can also talk about that. The DNA, you have that few. ACG is the same. Now let's talk about the differences, Right, first of all, the name. RY is RNA. Wow, W is DNA. The main difference is that why is RNA? Because RNA contains ribose sugar. Right, the name literally say ribose nucleic acid. Well, why is DNA? Because DNA has deoxyribose sugar. Remember, this is one of the example of their comparison. Deoxyribose sugar. Okay, and again, I believe this is simple how you have a lot of point in your nota. Okay, I'm just gonna put one more. Maybe you can say why is single linear. Why is just one line like that. Okay, but X and W is double helix. Again, you should have a lot of answer. You should have a lot more answer in your textbook, and not in your textbook, in your nota. I think you have a whole table with that. So yeah, you can go through that yourself if you want to. But yeah, that is some of the example as an answer. Okay, cool. Let's look at the last question. Some error occur during the process of transcription. So molecule Y is wrong. Okay, the RNA, the mRNA is wrong. Then what will happen to the uptake of sodium ion in root hair cell? Wow, okay. Suddenly you jump to this. Given that the protein synthesized is a transport protein, meaning that you are trying to make a transport protein, but it failed. Does that make sense? Why does it fail? Because of the wrong genetic code. So yeah, this is a typical table question that kind of give you a wrong scenario. You just have to, everything you learn is the same, but you twist it. You twist it to become wrong, to become fail. For example, you know that supposedly you are supposed to do what? You are supposed to do you see, when you already have RNA, you are supposed to translate into protein. Now you fail to do that. So wrong mRNA codon is translated into amino acid sequence. You see, initially it's supposed to be just this sentence, but now because the question said it's wrong, so you just have to add this one. You just add this single letter, this single word, then you get the answer, right? And similarly, the rest of the thing follow. So supposedly you are supposed to have condensation. Now, after condensation, you will form a wrong protein. You will form a wrong protein because you started with a wrong amino acid sequence. So finally, what is this protein? Transport protein. What is a transport protein that is involved in sodium ion? Remember, this is chapter 3. This is supposed to be carrier protein. So the carrier protein produced, it would be malfunction. Of course, I mean, if you use the wrong code, there must be something twisted. There must be something changed, something tweaked. So confirm you won't get the same thing. You won't get the original carrier protein. You might get a bit of cacat one or whatever lah. There will be some issue. And with that, what will happen? You confirm will influence the active transport. If the active transport decrease, so does the iron. So the sodium ion uptake will also decrease. So finally, don't forget, always, whatever answer you write, always link it back to the final point of the question. The question was asking for what? Outtake of sodium ion. So please answer that. Outtake of sodium ion decreases. Always remember to answer the question. Always remember, please, to answer the question. Because if you are too excited to whatever over here and you forget to answer the question, this will cost you 1 mark. The answer must be mentioned very clearly, or else the point of the question is to get the answer. Decrease or increase, and you write so many things and then you forget to talk about this, it will be a bit shameful because that is literally the main point. Okay, right, so make sure you always remember lah. After every question, check, have you completed the main point of the question? There must be a main ask, the main criteria to ask. So that one must be completed. Okay, that's it. All right, that's it for this question. Thank you very much.